All right. Hello, everyone. Welcome. Good morning, good afternoon, good evening, wherever you are across the globe. My name is Stoyan Stoichev. I'm part of the Resin Bio team. I'm going to be your host for today's webinar. Before we get going, just the usual housekeeping announcements. We are recording the session, and we're going to make it available for your offline viewing on our YouTube channel. So if you go to Resin Bio on YouTube, you'll be able to find this recording in about a week's time, and as well as all the recordings from the previous episodes that we merged. This is an open platform for discussion that's really geared towards emerging researchers. And so if you feel like you want to be part of it in the future, please drop us an email on emerge@resinbio.com, and we would like to definitely include you in part of the conversation in future episodes. Please post your questions in the Q&A section. And we also encourage live questions. So if you want to do that, so you can generate a bit of a discussion, please use the raise hand icon, and I'll activate your mic at the end of the presentation. So today's speaker is Assistant Professor Brian So. I had the pleasure of meeting Brian back in 2016 when he came over for a Skyline workshop in South Africa, and one of my first impression was how informative and engaging his lectures were on DIA, and I'm really happy that he's agreed to come and speak about his experience about data independent acquisition, which is really a technique that's become quite prominent and has enabled a lot of high throughput, robust applications to be possible with mass spectrometry. And of course, building libraries, extracting the data is a critical part of the DA workflow. And this is what Brian is going to be focusing on. Just a little bit of background about him. He's currently an assistant professor at the Ohio State University Medical Center Department of Biomedical Informatics. I suppose he had a little bit of a non-traditional career so far, where he started his academic career. He finished a degree in chemistry back in 2001, but then he stepped out of academia and he started his own company in data processing, data acquisition, called Proteom Software, which he co-founded with some of his colleagues, and then only came back to academia to complete his PhD back in 2014, I believe. That was at the University of Washington with the Mike McCoss lab. And so, Brian, welcome and good morning to you. Thanks very much for joining us today, taking some time off your busy schedule to chat about DA and spectral libraries.
Awesome, thanks, Stoyan. I really appreciate the introduction. I actually, while we're doing this, if people have questions and want to just pop in with questions as they go, feel free to invite them to speak. Also, if questions come up in the chat and I don't see them, then just blurt them out. It'd be fun if this were more dynamic. I definitely don't have enough material to talk through the entire hour, so it would be great to be able to do this interactively. Stoyan, is it okay if I put up some slides?
Yeah, carry on. I'll just stop share on my side.
Just making sure that I can do it. There we go. Okay, can you see slides? Good?
Perfect.
All right, so I guess to start, I've been coaching a lot of people through how to acquire data through DIA experiments, and almost always the biggest question that people have are how do I build libraries for analyzing my experiments, and how do I go about interpreting the data that I do collect? Because configuring the instrument is actually probably the easiest part of this. I think that one of the things that people have on their mind is how do they actually use this type of data acquisition. And so today I just wanted to talk about specifically about this one small piece of DIA experiments, which is building libraries for them and the sort of different ways in which you could do that. So just to before I get started, Stoyan mentioned almost all of this earlier, but just to disclose my relationships, I'm a professor at Ohio State, but I've also founded a software company called Proteome Software that makes scaffold. Most of the research that is presented here was funded by NIGMS and IARPA, with some of it funded a little bit by the National Institute for Aging at the NIH. Okay, so I first want to talk about data acquisition before we get into how do we interpret this type of data. And I think the way I like to think of DIA is actually like a targeted experiment. It's an experiment where you've selected in a for example, a parallel reaction monitoring, where you've selected a fragment ion mass. So for example, this particular blue mass at 548.3, and you've told the instrument to configure the instrument to constantly collect fragment ion scans across that. So you're looking at a distribution of or a set of fragment ion scans that you're monitoring across the elution profile of the peak. I think that this is sort of a really easy way to think about making measurements with, or fragment ion quantitative measurements with, mass spectrometry. The way that you typically interpret PRM data is using synthetic peptides. So you look at the distribution of the fragment ions and the relative intensities of the fragment ions you measure, and the shapes of those peaks, as long as the shapes of the peaks sort of fit what you expect the peptide to look like. And you see the right distribution of fragment ions, or the right intensities roughly, you can kind of make a call for it. And this is sort of analogous to a mass spectrometry based Western blot, but it's a little bit more sophisticated because instead of a single measurement, you're actually making, so maybe 4 or 5 or 6 different fragment ion measurements that are a little bit even more selective than a Western blot would be. So I think this is a really great technique that should be in your arsenal. But these stable isotope labeled peptides that you typically insert into these experiments so that you can monitor them, these are peptides that have the same chemical properties of the peptides you're interested in targeting, but they have a different mass because they have some additional neutrons added to the actual peptide itself. So they give you kind of an advantage in trying to interpret this type of data. So for example, this is a particular peptide, a phosphopeptide, that I had spiked in. So this is the heavy standard that's been labeled with extra neutrons. And I can see that it elutes at a very specific time point that helps me kind of identify that this is the right measurement I should be looking at, and that this other signal is actually an incorrect signal from an alternate positional isomer. This is a peptide that has the same precursor mass and many of the same fragment ions, but not actually the same peptide that I'm interested in. Okay, so how is this similar to DIA? I think of DIA actually as targeting masses in the same kind of way, but the masses are wider windows. So instead of targeting a very specific mass, so that 538.3, we're targeting 525 to 550, so a pretty wide precursor mass range. And instead of fragmenting one peptide, we're actively co-fragmenting multiple peptides at the same time. So we're seeing fragment ions for multiple peptides in individual mass spectra. This is an extremely complicated style of data to interpret, but at the same time, we're making measurements of the entire proteome as opposed to measurements of very specific peptides. But a lot of the same principles are important here. In particular, how we try to make these types of measurements and how do we detect peptides from this. So one thing that should be abundantly obvious is that we can't just do this with stable isotope labeled peptides. We don't have enough money, for example, to buy all of the stably isotope labeled peptides in the human genome that we might be interested in monitoring. There's just no way that we could do something like that. And so the way we try to deal with this is by detecting peptides using signals that we've already seen. But just to kind of touch on that, I just kind of want to show why we're interested in this type of analysis or this type of measurement, as opposed to doing a normal DDA style experiment. And I think one of the clearest things that we can see is that oftentimes we see peptides that have co-eluting precursors. So these would be peptides that have the same precursor mass, but are different sort of set of fragment ions. For example, this is a circumstance where we see an early peak that happens to be, around less than 30 seconds away from the peak that we care about, but it will trigger an MS2 in a region where we're not going to see fragment ions. And if we were looking at this with DDA, we might not even actually monitor this peptide, the peptide that we care about, because of dynamic exclusion. So this type of thing happens in all sorts of other ways. So for example, not just can we have co-eluting peptides that have the same precursor mass, but we can have co-eluting peptides that have different precursor masses. So here, the monoisotopic peak, that's that blue peak in the middle, follows a pretty normal distribution with the rest of the peptide, but the purple peak, which is the +1 isotope, and the red peak, which is the +2 isotope, are shifted over. And so that indicates that there is another peptide that exists at the same time and space, but again, a different precursor mass. And there are also circumstances that we see really good DIA signal for peptides, but we actually don't see any precursor signal at all. And there can be lots of reasons for this to be the case, which we could talk about at another time. But oftentimes this happens in circumstances where you're analyzing data with orbitraps or trapping instruments. Okay, so how do we interpret this type of data? So for example, this is a particular peptide that I'd be interested in from superoxide dismutase in a normal sort of human sample. And if I were to extract out all the fragment ions, this would be all of the B and Y ions that I could associate with this peptide, HHAA, etc. I would see lots of different signals across the entire time domain. And it's kind of hard to pick out which is the right signal to be looking at. But if you use computer algorithms, they can pretty easily say that, well, the peptide that you're interested in is eluting at 43 minutes here because I have very good commonality between the fragment ion signals. They all follow the same shape of the peak, and I can be confident that this is a good detection. So this is how you would normally detect peptides in DIA experiments, or easy to see peptides in DIA experiments, but it's not always the case. So here's another peptide in superoxide dismutase. These peptides are equimolar, so they exist at the same amount on column, but they actually have, or they should be equimolar in this case, because they come from the same protein in the same experiment. But this particular peptide doesn't show quite as clean signal. So it's hard to see where that might be. But if I zoom into the region where I would expect that peptide, it's hard to say, is the peptide eluting at 63 minutes or at 65.5, or is it eluting at 64? The answer actually is that it's this really messy signal over here at 65 minutes. And the only way that I can know that is I can use fragment ion intensities that I've already measured of this particular peptide that I've seen in a DDA experiment in this particular case. So if I've seen this peptide in a DDA experiment, I can say, well, this peptide specifically should have a nice Y8 and a Y9 signal, pretty good Y10, Y11, Y12, and I can pick those Y8 and Y9 signals as being high and the Y10, 11, 12 as being present in the background, despite the fact that there might be 20 or 100 different peptides eluting in this precursor mass range around this peptide at the same time. Obviously, this is an extremely complicated spectrum at the apex of the fragment ion space. Okay, and so by using the commonality of those ions, we can make a call as to whether the peptide is present or not. Okay, so that kind of like sets the story of why we want to use peptides in libraries, why we would want to use measurements that we've already seen to help us detect peptides that we haven't seen yet in a DIA experiment or in this specific experiment. And so the most common way to do this is with DDA. Just sort of like how I talked about, I had already seen this peptide here using DDA, and that made it much easier to be able to look at it with DIA. Okay, so that would be the easiest way, I think, or the most common way that people are involved in or starting to use libraries for analyzing DIA. There are some sort of advantages to this approach, but there are also some advantages to using alternate ways of generating fragment ion spectra. And one of the most common ones that is up and coming at this point is actually just predicting them de novo using deep learning engines. So I'll talk briefly about that, and then I'll talk about this approach to generate libraries using DIA data only. Okay, so before we begin on this, we should ask the question, what is in a library that we're interested in monitoring? So there's a couple of things that we want to capture in these libraries. One of them is the fragmentation pattern. So we've already talked about needing to use the sort of distribution or the shape of the fragment ions in the sort of analysis. So here we're seeing lots of different fragment ions that are associated with this particular peptide of interest, and that helps us make a confirmation for the detection. We can also use retention time information that's stored in that library. So knowing when that peptide elutes will help us make sure that the correct signal is visible when we're actually there, and then the other sort of major advantage that people don't sort of normally think about is actually you're looking at a fairly limited number of peptides. So typically in the library, you only have 4 or 5 or 6 potential peptides that represent that particular protein. Oftentimes, those peptides have very different intensity responses across that time frame. So for example, when we were looking at those 2 peptides from superoxide dismutase, one was very easy to see and it had very intense signal, but the other one didn't have very intense signal. And that's because it doesn't ionize quite as well. So we don't get quite as good signal from it, despite the fact that they should be equimolar. So the intensity response from the first peptide, the most intense peptide, versus the last most intense peptide, can span orders of magnitude. So here I'm showing you 3 orders of magnitude. So that's 1 to 1,000 down difference. Okay, so there's a question in the chat. So the logic of the library is that the fragment ions in DDA and DIA is the same. But the fragmentation suppression in DIA may cause a difference. So actually, this is a really important idea where it's important for search engines or these types of analyses to know that the DDA spectrum is not going to be exactly like the DIA spectrum. It needs to be close, but not so close. You shouldn't expect it to be so close because there are lots of different things that inform fragmentation. One of them, for example, is collision energy estimation. So with DDA, you know the charge state for the peptide, and you know the kind of collision energy that you should fragment that peptide at. But the measurement in DIA doesn't know anything about the charge state, and so the fragmentation patterns are still slightly different. So you can see, for example, in this particular case, I've got a DDA spectrum where I've got some peaks that are present, and they're present at sort of slightly different intensity levels. But as long as the broad strokes are good, then it's a good circumstance. Okay, great. So that's a great question, Gian. Okay, so how do we build these types of libraries? I think the one of the most common ways that people have been doing this is to use stable isotope labeled peptides to kind of help you adjust retention times between runs. So this would be, for example, run 1, which would be a DDA run. I could put into my experiment a bunch of, say, for example, these 8 different isotopically labeled peptides. They could be, for example, commercial IRT peptides, but they could be any type of peptide, to be honest. So oftentimes our lab uses PRTC peptides, which is a different type of retention time standard. But we know kind of something about those previously acquired peptides, and we've kind of lined them up on on some sort of arbitrary space in what we call IRT across the retention time. And then we detect all of these DDA peptides in red that exist across the entire gradient. Okay, so there would be a bunch of peptides that come from the actual run that we're interested in measuring, and we can plot those peptides on the same axis that we saw for our index retention time peptides. So the stable isotope labeled peptides, the ones we know something about already. And this kind of puts us so that we can put all of those red peptides on the same line and now assign those peptides what we call IRTs, or again, some sort of index retention time. And then we can go down to another run. So this would be, for example, our next run that we'd be interested in monitoring. This might be a different DIA run, for example. And we have the same stable isotope labeled peptides. We can put them on a same line, and we can say, okay, well, what are the peptides we see here, and how do they relate to the peptides we've seen in the past? And we can do that through this retention time line that we've drawn using the stable isotope labeled peptides. So by drawing connections between those elution times, we can kind of correct for where we should expect to see a peptide or not. So I think that's kind of a good strategy to try to deal with this type of problem. But we don't necessarily need to just use one run. We can actually do this for many different runs. So for example, we can take now all of the peptides that we've seen here in run 1, and we've seen a bunch of peptides that we've detected already, again, put onto that line. And now we've got index retention times for all of those peptides. So if we take all those peptides in a totally different run that doesn't have stable isotope labeled peptides, we probably detected some of those that existed previously. So some of those peptides are going to be really common to experiments. So like keratin, for example, is going to be in all of our experiments. But oftentimes, if you're analyzing, say, serum, you might see albumin in all of your runs, no matter what you're looking at. And you can use the albumin peptides in those cases to be able to draw your line of interest. And so that would help you kind of put in, so you now know where these particular peptides elute on this sort of index retention time line, and you can take all of the new peptides that you see in another run and put them on that same line. This actually helps you build a library. So here, for example, I've got all of these peptides that I've seen in my first run that had these retention time standards, and I can map them to the IRT line. And then I can take a bunch of new runs. So for example, this green run, I can detect all of those or some of those peptides that I saw in run 1, and I can map them to the same line now because I can draw a curve that's associated with that. And I can do that with another run, another run, another run. And I can generate sort of massive constructed DDA libraries just using DDA data that I've got sitting around. This is actually a really great technique to be able to mine through a bunch of different data that you've already got sitting around. Again, even if you don't have stable isotope labeled peptides put into the experiment. This gives you the ability to say, okay, well, this is where you should expect these peptides to exist on a retention time line. And this is actually how you can analyze data, new sort of DIA experiments without using IRT peptides inside of your runs. So there's a comment or a question in the chat by Jian Song. Just sort of like DIA neural networks doesn't use any IRTs or CIRTs. There's lots of other types of software that does the same. So our software encyclopedia does the same, Scaffold DIA does the same. They don't use IRTs because they use the endogenous peptides that you see in your libraries where you try to do like a preliminary run to identify peptides. And you can see a bunch of those peptides easily without having to worry about retention time. And then you use those as the anchors. So this is how you would draw that same type of line, again, using an experiment without having IRTs in your sample. You just use the endogenous peptides as that scale. This actually is a really great idea, I think, because IRT is a completely arbitrary space. It's a meaningless space that doesn't have any bearing on reality. So you can see that some of these IRTs are negative numbers even. Some of them can be extremely large. And It's just an axis to put things on. It doesn't matter what that axis actually is. So in some experiments, I've actually been trying to map a bunch of DIA runs to percent acetonitrile instead. So again, you don't really need IRT as the scale or as a yardstick. It's just a common yardstick that people use, or a common concept that people use. And you also do not need IRT peptides to be able to use the IRT space. Okay, great. So retention time warping can also be kind of a little bit of a challenge. And I think that this is something that we've been thinking a lot about, which is how do you draw retention timelines in spaces to do these kind of curve fits, specifically in circumstances where it's not a line, where you have more peptides than just a single set of IRT peptides. If you've got thousands of sort of retention time standards in your library, you might as well use them all and draw a really clean fit. And so we've actually been working on different types of ways to draw these lines through. And the approach that we've come across is what we call a kernel density experiment, where we take our coordinate space, so our sort of DDA library versus our DIA experiment that we're analyzing, and we divide it up into different grids. So we divide it up into 1,000 or 3,000 different grid points, like pixelation, and we try to treat each one of these peptides as a different sort of distribution. So we think of this as kind of like stamping down a distribution at these different points. And as they sort of stack up, we can see them sort of rise. So we can look at the shape of this retention time profile as those sort of like piles of stands stack up on top of each other. We can see this sort of clean shape across the retention time space. So even though, for example, there is one run in the library that is incorrectly calibrated, this doesn't sort of interfere with our ability to sort of trace the shape by walking down the ridges of this landscape of retention time that we're seeing here. So this gives us the ability to kind of follow the curve without getting distracted by some of these other ions. And this can be an advantage, a really nice advantage to our approach when we can use sort of these thousands of peptide measurements to draw a retention time fitting curve, whereas other types of approaches that use median-based methods or these high-performance IRT analyses that can kind of get tricked by incorrectly assigned peptides in your library. The kernel density is sort of like a really nice way to think about retention time fitting across thousands of peptides. It's extremely robust because it's just a very simple algorithm. Okay, so there are lots of these mega aggregated libraries. So I talked about aggregating libraries from lots of different DDA experiments all together into one massive kind of data set. That was actually done for our work here on Phosphopedia. This is a phosphopeptide library, but there's lots of different places where you can get good peptide libraries. So for example, there's the Swath Atlas, provides a bunch of different types of libraries. ChemData at NIST provides peptide libraries for lots of different types of experiments, samples, and species. There's a lot of these, and I think that for example, there's a new pan-human library that's being generated by Tian and Gao's lab. And I think that that's a really excellent resource. I think that one thing to note here is that it's important for you to be aware of the provenance of the library that you're looking at. So some libraries have more errors than others, and some libraries, for example, this ChemData NIST, don't include retention times. So knowing the library source and what the library contains can be really important for interpreting it. Okay, so DDA libraries are, I think, great, and they've been an incredibly useful resource for us for a long time. But there are a lot of challenges in trying to use DDA libraries. And one of them is that to generate a good DDA library, you have to use a lot of runs. So typically, if we're looking at sample-specific experiments, we would be looking at tens to 100 different sample-specific runs to analyze just a single organism or single sample. And if you wanted to do something for a pan-organismal experiment, sort of like the pan-human library, you need to look at like hundreds to thousands of runs. So for example, the pan-human library has over 300 runs in it, DDA runs. The phosphopedia library that we generated has over 1,000 different DDA runs, where we're looking at lots of different data across lots of different experiments to be able to identify as many possible peptides as possible. Okay, and the other aspect of this is that how you generate those peptide libraries is really important. So for example, if I did, and in this case, we did an experiment using SCX separated peptides, as well as high pH reverse phase separated peptides. This is in a yeast sample, so a relatively simple sample. And yet still the overlap between the number of detections that we see that are exclusive to the SCX library versus the high pH reverse phase library is sort of staggering, that the overlap is only about 40% between these libraries. That you really, if you want to build a good DDA library, you really need to acquire multiple sets of fractionation methods, fractionated peptides, using different types of fractionation approaches combined together in the same experiment. Okay, so Jian mentioned this a little bit earlier, but collision energy actually causes difference in fragmentation between DDA measurements and DIA measurements. And so the exact fragmentation patterns aren't the same. And this has to do with the fact that that for DDA, you can assume a specific charge state and you know what it is. And for DIA, you don't necessarily know what the charge state is. You have to fragment everything as if it's the same. I think the third important characteristic is that when you use a fractionation approach, so SCX or high pH reverse phase, you are separating out the peptide you're interested in monitoring from the background of all possible other peptides. And those peptides interact on the column, and that actually changes how they elute. And so we actually see circumstances when you're looking at peptides in an actual matrix, they elute somewhat differently than they elute in the fractionation experiment. So for example, this DDA set was run using a fractionation approach. So we generated a deep library. I think that this was the SCX library that we have below here, this yeast library. And this would be the retention times from the DIA experiment. We see that they're not precise, right? There's some fuzziness to the measurement. But more importantly, there's some error to the measurement, because in different types of fractions, these peptides will elute earlier or later, depending on what's going on in them. Okay, so there's a question from Azad in the chat. You touch on some great points here. With a lot of these sort of DDA runs, is it worth it to generate these DDA runs? And I think that that's a really good question. Is it worth it to build massive sort of like tens of injection runs for each of these different types of experiments, or hundreds of different runs for each pan-organismal experiment? And I think that that's something to weigh, definitely depending on what you're thinking about. But that's why we're going to get into some of these other approaches. So there are sort of two other different types of approaches that I'd like you to consider rather than thinking about building DDA libraries. One of them is to do peptide predictions using library engines like PROSIT. And the second one is to build your libraries using DIA data instead of DDA data. So that's a great question. We'll get to that right now, actually. So library predictions are a really excellent tool to be able to detect peptides using DIA experiments. So basically in these types of experiments, you just give a predictor like PROSIT, for example, the peptide you're interested in, the fragmentation, the charge state, and the sort of collision energy settings that you would use, and put that into a neural network, and it will spit out a fragmentation pattern. Same thing, PROSIT uses actually two dual neural networks, one to predict fragment ions and the other to predict retention time. And they work essentially the same type of way. We find that these fragmentation patterns are actually remarkably good. Like they're very they are consistently accurate with the type of data that we're interested in predicting. And they're essentially about as good fragmentation pattern-wise and retention pattern-wise as a DDA library would be. And so we've actually built a workflow to be able to just predict these PROSIT predictions from FASTA databases. So just using Encyclopedia, there's a convert setting where you can convert a PROSIT input from a FASTA. You can upload that data to the PROSIT web server, and then you can convert that PROSIT information back into a library here, going from a FASTA through to PROSIT into a DDA equivalent library. And as I said, the fragmentation patterns and retention time patterns are just as good as with DDA. So as I said, they're shockingly accurate. You can see, for example, the empirical data versus the predicted data for this particular peptide is spot on in terms of its fragmentation patterns. There are slightly small fragment ions that we don't see in the empirical data set, but the overall broad strokes are right on, even if they're not exactly on. So like Y9, for example, is slightly lower in the empirical data, slightly higher in the predicted. The retention times are similarly good. So for example, here is a retention time spread of about 1.5 minutes on a 90-minute gradient. That's usually enough to be able to pinpoint the accuracy of where the peptide should be. Oftentimes, this is about as accurate as running a DDA experiment right before your run is. Okay, and so we find that this can be a really good approach to be able to detect peptides. So for example, here I've got some yeast measurements on the left and some HeLa measurements on the right, and I can see sort of the number of detections that I can monitor in each of them. And so this is using Pecan, which is kind of like a benchmark. So this is using a library-free approach, kind of analogous to running sequest on the experiment. And then sort of this next one is using a combined SCX high pH reverse phase DDA library. So 2 different types of fractionation combined together into one giant experiment. So 16 runs over here built into the library, 39 runs built into the HeLa library because human samples are just a bit more complex. If we search that with a predicted library, we do pretty well, right? As I said, the fragmentation patterns are pretty similar, but we don't do quite as well. So there's a third step here, which is, as I said, the limited library actually helps you a lot. So rather than predicting every possible peptide in a FASTA database, the advantage of the DDA library is actually in looking at a limited number of peptides that you expect to see in your experiment. Because again, as I said, in this particular protein, this is a transcription factor Cas1. If I look at the top response, I see this is around E6, 5E6. And the bottom response here is less than 10E3. So there's over 3 orders of magnitude separation between these peptides. And you're much better off looking for peptides that are in this upper echelon. So peptides 1 through 10 versus peptides 30 through 40. And peptides 30 through 40, you're just not going to be able to see them in your experiment in most cases. And B, they're just going to distract you from being able to detect other types of peptides by sort of confounding matters, making you ask or look for more peptides than you need to, and making a mess of your statistics. This protein here is really common. So this was one transcription factor that we were interested in. Here is a map around over 700 different proteins, where we're looking at the difference between the highest responder and the lowest responder. On average, we see about a separation of 3.5 orders of magnitude between the top responder and the bottom responder in terms of intensity. So this is a really important factor in generating these libraries, is that oftentimes these predicted libraries just contain far too many peptides. That slows you down in terms of your analysis, but it also makes it harder to feel confident in the peptides that are really good measurements or really good matches. So this brings me to one important point, that if you're going to use predicted library approaches, and this would go in with something like DIANN or direct DIA from Spectronaut, in these cases, I would recommend not, if you're just doing normal PROSIT kind of library generation on its own, I would recommend that you look to narrow down the types of peptides that you look at. So rather than looking at all possible peptides, you should just look at the peptides that are likely to be present. So I recommend using a tidy database, a small database like UniProt, SwissProt, if available. So for example, this would be the number of peptides that are in the HeLa spectrum library versus the human FASTA, where we're seeing over about 1 million peptides of canonical peptides. These don't even include any miscleavages even. these peptides, so we're seeing a massive difference or a massive increase in the number of peptides that we need to monitor. But in particular, don't add in PTMs unless you're absolutely sure that they're present in your sample. So if you've done phospho enrichment, then you should look for phospho. But otherwise, you shouldn't look for phospho, for example, because phospho increases your search space by over 3 times. Again, you shouldn't look for semi-tryptic peptides or a higher charge space. In these cases, definitely less is more, and looking for just the canonical human proteins actually gives you the best sort of results here. Okay, I just want to spend a few minutes and talk about one last idea, which is gas phase fractionation for library generation, where instead of looking at one particular or looking at a proteome using sort of just one injection, a single injection, all of the experiments we've looked at so far have just been single injection. What if we were to inject that peptide or that sample twice? So we've got the peptide, it's just sitting in the autosampler. What if we just ran it a second time? But rather than just doing a technical replicate, what if we told the instrument to only look through a certain mass range? So for example, instead of in the full mass run, going from 400 to 1,000 and then repeating that like you would with replicates, if you were just to acquire from 400 to 700 in one acquisition and then 700 to 1,000 in another acquisition, you actually increase your confidence by having much narrower precursor isolation windows. So your actual measurements that you're making in these cases is much tighter, much more accurate. And so one thing to consider would be, could we expand this out even further? So for example, could we inject it 6 times instead of 2 times? And so here the idea would be you'd monitor from 400 to 500 in 4 MC wide windows, and 500 to 600, 600 to 700, et cetera, et cetera. So these are 4 MC wide windows. And if you were to use kind of like common techniques for demultiplexing, using, for example, staggered window demultiplexing, you can kind of shift over your windows by 50%. You can actually get this 2 MC wide measurement, or sorry, this 4 MC wide measurement into essentially broken into half into 2 MC wide measurements. And now you're looking at data that is the quality of PRM experiments without, but at the same time being sort of like PRM experiments, but for an entire proteome, because you're looking at 2 MC wide isolation windows, which is again the width of a normal DDA experiment or a normal PRM experiment. So this type of gas phase fractionation, I think, is really helpful for being able to detect peptides in deep experiments if you've got enough time to sample 6 acquisitions worth. Sudeep Ghosh asks in the chat, is it possible to design these gas phase fractionation experiments using ion mobility, like with FAMES? And it is definitely possible to use FAMES as an alternate to a quadrupole. In fact, I think one of the sort of original promises of tools like FAMES was that because they could act like a quadrupole if you didn't have a quadrupole in front of your instrument. Because you could essentially select on precursor mass. I think that one sort of challenge of trying to use ion mobility is that ion mobility is so commonly associated with precursor mass that it may not get you as much separation, orthogonal separation, as precursor mass would in these cases. It just does essentially the same type of thing as the quadrupole does in the front of your instrument. In many cases, but there may be some circumstances where trying to use ion mobility to do gas phase fractionation is a sensible approach, and then definitely is a possible approach to consider. But so getting back to this idea about doing these gas phase fractionation experiments where you've generated sort of a deep analysis of one specific sample, well, what if we were to use that sample for library generation? And because this is essentially the same quality as your DDA data or the PRM data, why not just build a library directly from these gas phase fractionated DIA runs? And so this, again, this idea is if you were to divide up the precursor MZ space into a lot of different windows, it would be much easier to be able to identify those specific peptides and then generate your library from those. And you call these libraries chromatogram libraries because they're generated based off of the actual chromatogram or the peak shape that you're seeing in DIA. And they give you a lot more sort of interesting information in these cases to be able to make these types of measurements. Okay, so here is a figure that's presented in Lindsay Pino's paper. She actually just jumped into the chat. I saw that. Thank you, Lindsay. On constructing DIA-only libraries. Where the idea basically is you've got a bunch of samples that you're interested in monitoring, these blue samples and these orange samples, and you create a pool of those, a mixture of those, this yellow sample. So you just kind of like take a sub-aliquot off the top of each one of those, or at the bottom of the well that's left over after you've run your sort of normal DIA experiment, and then you just acquire those one after another, where you add in your 6 gas phase fractions of your pool at some point during your experiment. You use those 6 gas phase fractionation experiments, those GPF DIA experiments, and you build a library searching a FASTA database or using a PROSIT prediction in those cases. And you use that library, that gas phase fractionated DIA library, to search your samples. I think the biggest advantage to this approach is that you haven't done any chemical fractionation. You've only done gas phase fractionation. So all of your column properties are identical. And the other advantage is that you're acquiring your gas phase fractionated DIA experiments with DIA fragmentation patterns. So you don't have to worry about the sort of fragmentation differences between DDA and DIA. So let me show you kind of what this means. So for example, if we're trying to do a DDA run, so this is a large scale DDA library that we generated. This is the 39 fraction DDA library versus our DIA experiment. We see kind of like a pretty good retention time fit that we can see relative to the sample that we're interested in, but it's not precise. And this has to do with all the fractionation we did ahead of time, that those fractionations cause slight changes in retention time. If we try to do that comparison, similar comparison against the chromatogram library, all of those points line up exactly on a line. And I think that that's remarkable. So instead of looking at retention time tolerances of 5 minutes, plus or minus 5 minutes in precision, we're looking at retention time tolerances of plus or minus 20 seconds in these experiments. And this absolute accuracy and the precision of the retention time, because we haven't done physical fractionation or chemical fractionation, we've only done gas phase fractionation post column, actually gives us a lot more confidence in our measurements. The other aspect of this is that fragmentation patterns are just simply more accurate, because rather than comparing DDA to DIA, we're comparing DIA to DIA. And so all of the assumptions that the instrument makes in both cases is exactly the same. So when we look at those sort of gas phase the deep DDA libraries using SCX or high pH reverse phase versus the predicted, the SCX and high pH reverse phase usually beat our predicted libraries. But if we use this approach to filter those detections using gas phase fractionation, we can actually get about the same results using both types of techniques. And I think this is kind of really important here, is that no matter sort of what strategy you use, whether you build these libraries or whether you use a prediction library, as long as you collect 6 gas phase fractionation DIA libraries, you can build not only larger libraries, libraries that are essentially complete for that experiment. They represent the total proteome of that experiment, the total detectable proteome for that experiment, using the sort of settings that you've got or the systems you've got. And that gives you an idea as to what the entire space is. Whether you're using DDA as the background or these predicted libraries. And as a result, we find that these predicted approaches combined with gas phase fractionation, what we call empirically corrected, where we can correct the prediction based off of our gas phase DIA runs, actually makes for the best type of, or the best type of library given the sort of amount of time that you have to make those measurements. So with that, I'd just sort of like to conclude. We talked about library generation. We talked about the strengths and weaknesses of predicted libraries. And this gas phase fractionated DIA experiments, I think, are the sort of what we need to do for going forward. So with that, I'd like to acknowledge a bunch of people. So I mentioned Lindsay earlier. A bunch of this work was done in Mike's lab, and Damien for help more recently. So with that, let's let's have some discussion about this. I noticed that there's a couple of points in the chat. I'll try to answer these right now, but then I'm going to close down the presentation, and then we should have sort of like some comments for the rest of the time. I guess there's not a ton of the rest of the time, but let's continue this. Yeah. So Ivan asks, is it sensible to predict good flying peptides for each protein? There have been approaches to do this. We've actually tried to do this with a tool called Prego. It's very hard to do it. There may be some deep learning based approaches. So far, there hasn't been a lot of success in this type of realm, but I expect there to be somewhere soon.
So Brian, there was also a question before that from Stefan about gas phase fractionation suffering from ion separation. It kind of turned into a thread because there's a question later on in terms of from Jared saying, would you combine GPF library fractionation with kind of normal fractionation? And would that is that worth going deeper given the extra mass spectrometry time to account for the ion suppression with GFP fractionation? GPF, sorry.
So I think those are great questions. And I think that if you are going to fractionate your normal samples, so for example, if you want to cut your normal samples into for example, soluble and insoluble proteins, and then analyze each of those independently, for every one of your normal experiments, then you should definitely do the same thing for your library as well. But I would argue that there's not really a point to doing that to build a bigger library if you're not going to do it for your actual samples. Because if you cannot detect the peptide using your gas phase fractionated libraries, you're never going to be able to detect it in single injection DIA, because the constraints of the gas phase fractionation versus sort of single injection DIA is just, it's the same data, it's just higher quality kind of thing. There's just no way that you would be able to identify consistently a peptide in a single injection run that you could not see in your 6 gas phase fractionation run. So putting more effort into the 6 gas phase fractionation run to build a deeper library is essentially going to be meaningless in those cases, if that makes sense.
Okay. And then
I think that's an interesting idea, though. But if you are going to do sort of an example like soluble versus insoluble fractionation ahead of time for every one of your samples and analyze each of your samples in twice, then I think it would make sense to build essentially 2 different libraries, one for soluble and one for insoluble.
We had a question from Lindsay on FAMES and about CVs when acquiring DI data. Does that affect how your actually single injection data is then acquired?
Yeah, so I guess again with FAMES, similarly, if you're going to use FAMES in your single injection runs, then it would make sense to do I think different FAMES CV libraries, sort of one after another. But I think that should be something you should ask: is it sensible to acquire different, or acquire your DIA runs with sort of multiple CV settings or multiple CV windows versus just acquiring it with more precursor isolation? Because you have to spend time collecting those scans. Is it better to acquire 2 scans where they're separated on CV, or is it better to acquire 2 scans where they're separated on precursor isolation window using the quadrupole as a different sort of separation? I think that both of those strategies are, I think, worth trying to play out, but I suspect that because the quadrupole is just significantly more precise than the FAMES CV estimation is. We know exactly what the masses are for these peptides, and we can use the quadrupole with very high precision. It would strike me as being unlikely that you would get great gains using FAMES independent or instead of the quadrupole as for separation. I hope that answers your question, Lindsay. It's a great idea.
I think we also had some follow-ups on that. Let's see. Trying to go in.
So Benny and
Benny.
Lindsay mentioned, is it worth trying to have, particularly if you've got specific peptides that you know are going to be interestingly separated with CVs that you can't separate using just the quadrupole, for example, phosphopeptides. I think that's a circumstance where there is a potentially some sense in trying to use FAMEs in addition to doing a normal quadrupole experiment. That said, we need to start including collisional cross-sectional measurements inside of the libraries that we're making, which it's been a struggle to get people to even put retention times into the libraries that we're making. So for example, massive KB doesn't have retention times, or the NIST library doesn't have retention times. I think it's going to be some time before we're going to get collisional cross-sectional measurements, but I look forward to being able to get those types of measurements going forward.
We have a question from King Zhang about the optimal GPF window. If you go too small, is there an issue, or is it like Dalton's like the optimal size?
You're absolutely right. There is an optimal size, and we find that 6 injections is about the optimal size. If we've done experiments where we've increased it out even further, where the sort of increase in precursor isolation window is offset by a decrease in signal. So as you sort of get smaller and smaller, you start splitting isotopic packets around a peptide more and more frequently. So where your monoisotopic peak falls in one window and your +1 peak falls in another window. And that can be extremely problematic to try to reconstruct. There really aren't tools to be able to do that in a sensible way. To look for those types of measurements where you look for monoisotopic in one set of windows and your +1 in another set of windows. Right now, there's just not a good way to do that. But there's some options, I think, for people to try to think about how they could do that type of work going forward. So from our experience with normal data acquisition methods and normal software, going from 4 to 6 is a benefit, but from going from 6 to 8 or 6 to 12, you usually don't get anything for your efforts, and sometimes you actually lose something because of those split isotopic measurements. That's a great question, though. Yeah.
We have a question from Jiang about, do you think that there are DI detection limits of HeLa with 1 hour gradient? What is the DI detection limits of HeLa with 1 hour gradients in precursor/peptides?
That one is a hard question to answer because instruments are changing as we go forward. So for example, a lot of the measurements I showed today are done on a QEHF. If you do the same types of instruments on an Eclipse or an Exploris, you actually get significant sensitivity gains because of the ion optics. And so it is possible to run those instruments somewhat faster and get higher number of detections. And I suspect that we will continue to see these types of optimizations from the different instrument vendors. I think that reliably, the sort of limits in trying to monitor HeLa in an hour for these types of experiments is going to top out somewhere around, if we think that there might be 12,000 total expressed gene structures, there might be at most 8,000 to 10,000 protein groups, I think, that we'll be able to detect from an hour gradient. And I think in today's day that it's hard to get above 6,000 in a 1 hour gradient type of experiment in this type of construct. So hopefully that kind of gives you some idea as to sort of what we're shooting for in measuring HeLa or those types of cell lines versus where we're actually at. So there might be 12,000 different types of proteins that might be in a sample based on sort of RNA expression levels. And maybe we can find 6,000 in a 1 hour gradient, maybe 8,000 in a 90-minute gradient kind of thing.
Okay. Maybe a follow-up question from my side. You highlighted how accurate the prediction process libraries are. How good are they across platforms? So a lot of the work was done on orbit traps, and were they as good as on top instruments in terms of?
That's a great question. Yeah. So actually, like in just like how in Orbitrap instruments, we get really good detections using the pan-human library, which was generated using QTOPS, SciEx QTOPS. I think that the PROSA predictions are actually quite good once you dial them in for what it looks like on your type of instrument. So one of these important characteristics that PROSIT requires to use, and I think that any good quality deep learning engine for predicting fragmentation uses, is some kind of measurement for collision energy. So PROSIT uses NCE based off of the Orbitrap NCEs. Tuning NCE is really important. Even if you have a Orbitrap instrument, NCE means something very different depending on whether you're using one instrument or another instrument. Two identical instrument makes, just different delivery dates, will actually have different NCE calibrations. So if you use sort of example PROSIT to predict peptides for your TOF instrument, I would take a DDA run and I would go through and do a NCE calibration experiment to estimate what sort of the equivalent NCE configuration for your hardware is on the TOF instrument, or what is the closest NCE configuration, and then use that for your predictions. And I think you'll get more accurate predictions than just sort of going in blind without that type of information.
Yeah.
Does that make sense?
Yeah, absolutely, absolutely. Okay, so we had a little bit of a discussion between Jared and Lindsay here. I think it carried on from the previous comment in terms of whether it's worth doing like fractionation combined with the GPF, and so one of the comments is that if you're thinking of enrichments, that you want to have the fraction that you actually are looking at. So if you're looking at nuclear proteins, you want to enrich them and then do your gas phase fractionation, so you can actually look at the representative proteins. Is that something that you would advise as well?
I definitely would advise that. So if you're interested in looking at nuclear proteins, for example, you are going to enrich for nuclear proteins in all of your single injection runs, then you should enrich, you should do that same enrichment, that same fractionation for your gas phase fractionator runs. However, if you're interested in nuclear proteins and you do not do nuclear fractionation for your single injection runs, I would not do the nuclear prep for your gas phase DIA library generation, because the background, the retention times caused, or the retention time shifts caused by the background changing or the matrix changing are fairly significant in terms of your ability to estimate what that is accurately. And you might get shifts as much as a few minutes. Whereas if you were to identify your nuclear proteins out of your unenriched gas phase fractionation runs, you would get estimations of around 20 seconds or so. So I think that it's important, however you collect your single injection DIA, all of the processing steps you do for it, you should do all of those same processing steps for your gas phase fractionation DIA.
Yeah, this makes sense. This is the same concept with SCX or reverse phase fractionation, where you're changing the retention times because of the matrix effects. I think this kind of let's see if everything is gone through everything. If anybody has more questions, if you want to raise a hand, ask a live question, you're more than welcome to. You can carry on with this. I don't know, Brian, how's your time? But
yeah, I'm happy to answer more questions. Jarrett brings up a really excellent point that you should keep your gradient length the same for, again, just like with what we were talking about earlier. Gradient length actually changes the peptides that each peptide interacts with in the background. So in a sense, gradient length doesn't change the matrix, but it does change kind of like the local interactions. And we do see retention time shifts. If you switch from a 90-minute gradient to a 60-minute gradient, there are retention time shifts. So rather than trying to collect like a 4-hour gas phase fractionation DIA library to get the deepest DIA library, you should just collect a 60-minute gas phase fractionation DIA library if you're collecting 60-minute runs, or 30-minute gas phase fractionation DIA library if you're collecting 30-minute runs. Does that make sense? I think that's a great point.
And it turns the whole concept around where initially we learned about IRTs and then we can actually acquire data across different gradients and normalize everything with IRTs, but that doesn't really account for those local environment changes that you just described.
Yeah, so in our lab we don't use IRT peptides anymore because we use them for calibrating the instrument, making sure that the instrument's running correctly. But we do not put them into our samples because we have thousands of endogenous peptides that we know where they should be in those samples already. And so there's no point in looking specifically for IRT peptides versus your endogenous peptides, because you will definitely see 900 of those 1,000 peptides you expect to see in every one of your samples. And they can be sort of your retention time calibrator rather than the IRTs that you pay for.
Yeah, I agree with you completely. They could be used for system suitability, but not really so much for retention time because you have the endogenous ones.
And that IRT space, I think, is arbitrary, right? It's completely arbitrary. It can go into negative numbers. It can go skyrocketing high. It has no grounding in reality, the IRT space. So you could use any type of retention time calibration space. You can calibrate to another run. So if you've got another run on your gradient that you set between 0 and 90 minutes, that can be your retention time space. You can calibrate everything into that 0 to 90 minute space. Alternatively, you can calibrate between 0 and 40% acetonitrile, depending on what your percent B is on the column. So the actual IRT space isn't really necessarily useful when you're doing things in your own lab. The only advantage to looking at IRTs is that other people have mapped their peptides into IRT space. IRT, but because it's just sort of like an arbitrary concept, I think it would make a lot more sense for us to start mapping peptides into a percentage acetonitrile instead, for example, because we all use acetonitrile as percent B and C18. So if we're doing retention time with C18, we might as well just do it to something that is we all have as a common reference point.
Yeah, this makes sense. Okay, I don't see any more questions. Last chance for anybody to raise anything. So Jared says that you can come by his SMS poster. Yeah, we would love to see that, Jared. For those that are lucky enough to get there, we will definitely see it otherwise online. So I don't have any more questions. So I just want to thank you, Brian, for a really, really insightful presentation. I definitely enjoyed it and learned a lot once again. And yeah, so as I